So I'm very happy to introduce our final invited speaker, Dr. Melanie Stefan. She's now at the medical school in Berlin. She was until very recently at University. Was it University of Edinburgh or was Area 12?
Was University of Edinburgh. Yeah.
University of Edinburgh. I knew it was in Edinburgh, but I wasn't, for a moment, I kind of blanked out which one it was. Melanie has worked a lot on cooperativity, signaling. I think CaMKII, we probably will see some of those topics appear in her talk. And we're very happy to have you here, Melanie. And let's get started. Just let me know when you need to advance the slides. So
Fantastic.
Okay.
Okay, thank you very much. Thank you, Pedro, for having me. This is a really great opportunity and a really great meeting. Could you advance to the next slide, please? So this is really just like introducing myself once more. So my lab is working on memory and cognitive function mainly, and we're doing this by using molecular pathway modeling a lot. And we also do a little bit with big data from sort of individual learning platforms. So I've been doing a lot of modeling, and I'm doing a lot of it in CaMKII, and I also teach CaMKII to my students because I think it's an excellent tool to sort of gain intuition for biological systems. So I was, as Pedro said, for the last 7 years, I was based at the University of Edinburgh, but also I traveled to China several times a year because I had an appointment at Zhejiang University in China to teach. And so to share some of my China experience with you, and I swear this will become relevant, we'll start with a little Chinese lesson. Yeah, thank you very much, Pedro. So there's a little Chinese character here. And I mean, this usually works better in a classroom where I can sort of see people reacting, but just note to yourself, do you know what it means or not? And does anybody know what it means? Would anybody like to volunteer an answer? Okay, yes, it's a tree. Excellent. Thank you, Caroline. Cool. So we know that this is a tree. Can we have the next slide, please? And can we have the next one? So, okay, so now we see another character. Do you know what that means?
It must be a forest, no?
Yeah, it is a forest, exactly. Very good. Yes. So hopefully there are some of you who did not know the tree character, but knew the forest character, thus proving that Chinese is very easy and that you can learn it within minutes, because a minute ago you didn't even know what a tree looked like, and now you know what a forest looked like. And what you did to put that together was you basically just said, okay, now that I know what a tree looks like, a forest is just a bunch of trees, right? And this is a very important to later. Case in modeling. Can we have the next slide, please, Pedro? And it usually goes like that, right? I say, okay, we want to build a model, but we have very little data to constrain the model. And then other people, especially if you can decide, don't have enough data. How do you know that your model isn't really good? And on the other hand, if I say, well, I have all the data about system X, I can build a really, really good model because they have, what do they say? They say, well, if I say, so today we're talking about that a little bit. So Melanie, if I know everything about the system, yeah.
Your connection is coming in and out. I don't know if you can move around to a place that has better connection.
Okay, let me.
Maybe you can turn off your video.
Okay.
Yeah, maybe if you turn your video off, that will be okay too.
Excellent. Okay, so I turn my video off.
Yeah. Okay.
And I've moved to the other side of the room. So let me know if this is maybe better. Can you hear me now?
Yes, we're hearing you very well now.
Okay, cool. So basically, so if I say we want to build a model, but we don't have enough data, people say, well, then your model won't be any good because you don't have the data to constrain it. But on the other hand, if we say we want to build a model and we have all the data, we can constrain the model really well, then people will say, well, but if you have all the data, in other words, if you know that much about the system already, what possible thing can you learn from building a model? What is there to learn from a model if we already have all the information about the system? And so what I'm trying to convince you about today, which I think some of you don't need that much convincing, but maybe some of you need a bit more convincing, is that even if we know a lot about a system, it's still very useful to build a model because models can basically surprise us and show us things that were not immediately obvious. So yeah, you can go on. We will look at the first example. And the first example, I call it Exhibit 1. And it's really like a very classical model. It's 100 years old. Can we see the next slide? And it's the Lotka-Volterra model of a predator-prey system. So it works like this. We have a prey animal, in this case, a bunny rabbit. And this bunny rabbit what do bunny rabbits do? If anybody has had one as a pet, one thing they're very good at is they make more little bunny rabbits. Can we see the next slide, please? Yes, so they reproduce. And the other thing they can do, unfortunately, if you can see the next slide, is they can die. But bunny rabbits don't usually die of old age. They are eaten by foxes. Can we see the fox? Yeah. And the foxes can also reproduce. And the bunny rabbits help in fox reproduction, not in the sense that they, I don't know, artificially inseminate foxes or anything, but in the sense that the more rabbits there are to eat, the stronger the foxes, and the more the foxes can basically reproduce. So their reproduction depends on the rabbits. And then foxes do basically in this model die of old age at some point. So this is a super simple system. We only have basically two participants. We have the foxes and the rabbits, and we have like very few sort of events or processes that can happen. We can now add little parameters to all those arrows. So basically say how quickly these things happen. We call them alpha, beta, gamma, and delta because we are very creative. And then we can basically write the system. Can we see the next slide? Like a system of basically reactions, right? So for instance, a rabbit becomes two rabbits with a rate constant of alpha. When a fox and a rabbit meet, only the fox is left with a rate constant of beta, and so on and so on. So we can sort of, and you can, if you want, take a minute to convince yourself that this is basically the exact same model that I have shown you before. So foxes and rabbits with a different kind of rate constant, their meeting will lead to more foxes being produced, and the foxes can die. And now what we can do next, if we can see the next slide, is we can basically build that into a system of ODEs. Thankfully, CaMKII does that for us because writing ODEs by hand is hard. And so then we can basically run a simulation. So I would like everybody to maybe take half a minute and think about what would that look like if we start with some number of foxes and some number of rabbits. Can you predict where the numbers of foxes and rabbits would go as time basically progresses? And feel free to like post in the chat if you have thoughts on that. I can kind of see the chat here when someone posts, I think.
We don't seem to have any answer yet.
Yeah. Oh, we have Kyle.
Oh, there's some of them, yes.
Yeah. Kyle, Theo, okay. So we have the word oscillation has been mentioned. Caroline says very correctly that it depends on the rate of reproduction. But in a healthy ecosystem, they would stay constant. So you can imagine a scenario where basically rabbits are replaced as they're eaten by foxes, and foxes basically reproduce and die. Both of them reproduce and die at rates that are such that there is an equilibrium and that both populations stay more or less constant. And that's true, that's one possible outcome. And then if it gets too low, that's a good point, Theo. If it gets too low, then it could collapse, and then both populations would collapse, right? If we have 0 rabbits at some point, then there is 0 to eat for the foxes, because in this model, all the foxes eat is rabbits, and then everybody dies. It's basically like Hamlet. But so you can also, so there's different scenarios, right? You can imagine the everybody dies scenario. You can imagine the, oh, it just evens out at some equilibrium where basically the 2 populations are constant. And there's a scenario which we're going to show now, where that some of you have mentioned as well, where you have oscillations. So if we can see that. So that comes from actually a capacity simulation. So we have the foxes in red and the rabbits in blue, and we can see that, and you can sort of think through it. So when the rabbit population goes down, the foxes don't have anything to eat anymore, so the foxes die. As the foxes die, the rabbits can grow again. As the rabbits grow again, the foxes have more things to eat, so they grow again, and so on and so on. So it sort of oscillates around. So I want to draw your attention here to, it looks like it goes down to 0 every time, but we're looking at densities here, not numbers. So what it goes down to is a density that's very small, but not exactly 0. So this is why the system sort of can always come back and recover. If we had literally 0, right, there would be no coming back from that, as Theo, I think, said. Fantastic. Now, this is kind of something that maybe some of you have seen before, and maybe some of you were able to kind of just think through and imagine, right? So this sort of, that you get some kind of maybe cyclical behavior, maybe that's not completely counterintuitive for some of you, although it's also not completely intuitive, I think, if you see a system like that for the first time. Is this actually just, you know, our model doing crazy things, or does it actually happen? If you can see the next slide. So this is now actually a famous study of lynx and lepus, whatever. Is that
wolves.
No,
is it wolves?
No, lupus is wolf.
Okay.
Anyway, 2 American species of animals, one of which I should really have prepared for this better. I knew this at some point. I think it's a, yeah, I will, oh, a snowshoe hare. Yeah, thank you, Theo. Okay, good. So we have the snowshoe hare that is being eaten by the lynx, and we can see their populations oscillating in kind of a similar way that we saw in the model. And it's kind of really nice because, you know, you would think the model is maybe, it's super simple, right? It's super simplified. It pretends there's only 2 species in the universe and they only interact with each other. There's no other environmental influences. It's really simplistic. But it is able to predict a behavior that we can see in some instances in the real world as well, which is, I think, really nice. Now let's take it a step further. Can we see the next slide? And let's say there is a country, let's say country A, that has a problem with its rabbit overpopulation. And those rabbits cause damage to agricultural crops. And the prime minister of country A has heard that foxes eat rabbits, and because country A, which shall remain unnamed, but it starts with A and ends with Australia. This country has some experience with introducing, so the prime minister thinks, and that will control the rabbit population. So the question is, will that work or not? Any thoughts? So I mean, one thought is, well, how often has this worked for you in the past, Australia? And the answer is not very often, and most of the time it didn't work because the predator species that was introduced had other side effects. Basically, it was destroying the crops even more, for instance, or it was reproducing uncontrollably and then become a pest by itself or something like that. So there's other environmental factors that need to be considered. But even within the model, if we look at the before, so that's the model that I showed you before, where we start with 70 foxes, I think, and 100 rabbits. But if we now say, okay, we introduce more foxes, can we see the next slide? What you see here is really interesting. So we doubled the number of foxes, right? We started with 150 foxes here, same number of rabbits. You can see the oscillations are slowing down. So in a way that's good. So we spend more time rabbit free or with very few rabbits. But what you also see is that the peak goes up much higher than it did before. So in the simulation we showed before, we had a peak of around 100 rabbits. And now we have a peak of around 200 rabbits. So the y-axis changes here. So we made the problem more rare, but we made it much bigger. Right? We doubled the size of the rabbit problem. And that probably not what country A intended. So this is really, and I think this is really where it gets counterintuitive, because we would think usually, well, if you introduce more predator, you get rid of more prey, right? So more foxes would mean fewer rabbits, but it can actually have the opposite effect, not immediately, but in a later sort of phase of the oscillation. So this is something where the model really shows us something that I think, I mean, to me at least, was unexpected, and I hope to some of you as well. And where really, I mean, it's such a simple model, right? It's a model that's 100 years old. It has very few, let's say, reactions or equations, but already it shows us something that we couldn't have just guessed, or at least we couldn't have guessed it easily. So this is sort of my first argument, my first exhibit. Can we move on to the next slide, please? Oh yeah, okay, ta-da, fireworks. So this is what we call emergence, right, or emergent properties. And it's one of my favorite things is when something comes out of the model and surprises us. Okay, switch gears a little bit and talk about diagnostics. And first of all, I want to again sort of introduce a little exercise or a little game in this case. So can we see the next slide? And this is connected with the fact that I just moved to Germany last month. And so now I have to become interested in German football, or soccer, as you call it in the US, which is wrong, but fine. So what we do, at least in Europe, and I think in other places as well, when a big football tournament comes up, is we have those little cards with like footballers' pictures and names and stats on them. And then we can go to a shop and buy like a pack of 10 of them. And we can try to assemble collections of them, right? So let's say, for instance, I have a big shop in town. I want to buy all the Panini cards from there. And I want to get a full German national team collection, basically. So can we see the next slide? So we can imagine that if I do that on my own, like I buy all the cards that are in the shop, right? There's like maybe a big stock of them. Then it's quite easy for me to assemble this collection of all German players. If two of us play, let's say Pedro and I both play the game, we both try to get a full collection of German players. We buy all the cards that are in the shop, we divide them between us. It's still quite easy for both of us to get a complete collection, therefore to win the game, right? But what if we increase the number of players higher and higher and higher? What do you think would happen to the number of winners?
It depends how many cards there are of each player. If they didn't make some of them more rare than others.
Okay, let's say there's, I mean, that's a good point, but let's say there's an equal number of cards of each player. Does it matter? Actually, Petra. I mean, it matters how many winners there can be. That's true.
Yeah, yeah. Well, it's a good question. I'm not sure.
Mm-hmm. I'm increasing the number of football players or card players. Yes, I should have said that. Sorry, this was not well explained. We are increasing the number of people who play the card game. So we're increasing the number of collectors. It would decrease, Katie, yes. So, and I think this is something that at first is not completely obvious, but if you have so at first, right, if you just, let's say you have a ton of cards and you have only a few collectors, at first, the more collectors you have, the more winners you have, because every collector becomes a winner. And then you would maybe think, I mean, not Katie, but I think many of us would think, or I at first would have thought that at some point it just like it evens out, right? So if you add more and more people collecting, you can still only have, let's say, 20 winners or something. So you just like reach a plateau of how many people can win, and that's that. But you don't reach a plateau. It actually goes back down. The number of winners goes back down, because if you imagine you have like let's say in the most extreme case, let's say you have like 100,000 cards, but you also have 100,000 players, then nobody can win, right? Because everybody gets one card and you can't have a collection with one card. So the more players you have, the more players compete with each other for those cards and to assemble a full collection. What could you do? So how could you modify the game to create more winners?
Where you produce more cards?
You produce more cards? What about with the same number of cards?
Below exchanging of
uneven
cards.
distribution.
So again?
So different players can get different number of cards.
Different players can get different numbers of cards. Yeah, yeah.
That players
And what, I mean, I don't know if any of you
exchange cards.
exchange cards. Okay, excellent. Yes. So this is what we did in the schoolyard. At least I did that, right? Like I said, oh, I'm only missing one player to get my full set, right? Could I exchange? Yeah, maybe I have two of another player. Could we exchange so that I have the full set, and then maybe, you know, you can use that card that you exchanged mine for to build up your collection. So if we exchange cards, then we at least then there is, if we exchange sort of in a clever way, if we collaborate, then there is a chance. It will still not like continue to increase linearly, right? Because there's just a limit to how many complete sets there are, but at least we can get as many winners as there are complete sets. And this is because we have cooperated on our card game. And so in case you were wondering what that's all about, this is actually what happens a lot in biochemical system, because quite often you can think about a biochemical agent, a protein, for instance, that binds to several other ligands. So it is basically a collector of ligands, and it is maybe activated or something if it is fully bound, right? So for instance, Calmodulin, which I work on a lot, a really important protein, not only in the brain, but also in the brain, it is activated by binding to 4 calcium ions. So it has to collect 4 calcium ions in order to get activated. And with a system like that, you would think that if you increase, for instance, the calmodulin concentration, you would get more active calmodulin. And that's true up to a point. But then once you are over that point, you get less, not only does it does it sort of plateau out, but you get less active calmodulin as you increase the calmodulin concentration, because of what, I think, was it Maria who said that earlier? Because of this sort of collecting problem. And one thing that actually helps is cooperative binding, right? Cooperative binding means that if a calmodulin is already bound to 3 ions, it's actually more likely that it will get bound to another one, sort of the last calcium increases. Whereas it's more to bind more calcium. So basically, those who already have collected a lot, I give them collecting items, and they say about or they lose them basically. So it's kind of a winner takes all kind of, it's not entirely winner takes all, but like winner takes more, right? So and it's the same game, right? If I say I already have 10, can I trade one of your cards for my school lunch or something? That's a stronger argument than if I say, oh, I already have 3, I only need 8 more to make up my complete set, right? So there's a sort of aspect of cooperativity here.
Melanie, I'm not sure I'm on the right slide. I'm still on how to win a game.
Yes, yes. We're still there, yeah.
So the difference between the collectors and the cooperative collecting.
Yes.
Okay.
Yeah.
Because your connection sometimes goes a little slow,
okay.
But now it's okay.
Okay, cool. So what I'm saying is that this cooperative thing is really important in biochemistry as well. And one example for that is a diagnostic test, because if you think of that, I mean, I used to have to explain this. If we can see the next slide. I used to have to explain how these work and how these look, but now, like, you've done this probably a million times in the last 2 years, because this is how the lateral flow tests for COVID also work. So if you basically, the way a lateral flow test works is you have your antigen that you want to detect, and the antigen is basically the collector. And it has to collect 2 kinds of antibodies. So first of all, it's labeled with a labeling antibody that makes it show up in color. But we don't see it usually as it just like diffuses through the membrane, because it's too dilute, right? So even if it is actually labeled with that color, or with a fluorescent label, whatever, or gold, we don't see it because it's so dilute. So we need something to concentrate it. And the way it's being concentrated is that on the test strip membrane, we have basically a stripe that is full of capture antibodies. So the antigens that have been labeled will bind to those capture antibodies and will basically concentrate themselves along that narrow line. This is how it shows up as a line on the test. And the antigens that don't bind to the capture antibody will just diffuse out of the test strip, so we won't be able to detect them. And the antibodies that don't bind to a label and just the capture antibodies will be invisible to us, right? Because they are at the right place, but they're not labeled. And now, because the antibody binds to 2 different antigens, we can think of the antibody as a collector, and we can think of a win, right? A win is when we see this particular antigen, and this only happens if it has a full connection, if it is bound both to the capture antibody and to the label antibody. And we can also now, from our vast experience cards that we now have of the, you could have a really unfortunate situation where the antigen competes with other antigen molecules for those antibodies, and you end up with a lot of antigen that is either bound to just a labeling antibody or either bound to just a capture antibody, and therefore will remain undetected. and this has been shown with malaria tests, with at least some brands of malaria tests, is that if the malaria titer is quite high, the antibody type, the antigen titer is quite high, then you can actually get a false negative result. And then if you dilute the sample though, you can get a positive result. So this is sort of the how the high dose, it's called a high dose hook effect. And it's a known problem for some pregnancy tests as well. So if someone is too pregnant, they may show up as not pregnant, unfortunately. So this is really something that has a true sort of application. And that, again, I think is not maybe immediately clear if we just think about how a diagnostic test like that works and what the parts are. But modeling something like that, for instance, in CaMKII, can show us the kind of curve that we saw on the last slide, can show us how it goes up first and then goes down. And then we can use CaMKII to, for instance, compare different kinds of tests, right? Is the type, if we can create diagnostic tests against different types of antigens, where the type of antigen basically can we choose it such that that problem is reduced or mitigated. Okay, can we see the next slide, please? And finally, I'm talking about kinase inhibition, and this is really where I'm sort of talking about my core research interests in neuroscience. So if we can see the next slide. So what we work on a lot is the molecular basis of learning and memory. So if you have had a bit of neuroscience, you can, maybe even if you don't have had, if you haven't had a lot of neuroscience, you know that we remember things and we can learn things. And the way that memories are stored in the brain is through long-term potentiation of synapses. So a synapse, as a connection between two neurons that is repeatedly activated, gets stronger with time, and this is what we call long-term potentiation of the synapse. And we can see that in sort of an electrophysiological setup on the right. So we have a synapse being repeatedly triggered or repeatedly activated, and then what we plot on the y-axis is basically the response. And for most synapses, that's basically stable. So you activate it, it will respond. You activate it again, it will respond again with the same sort of with the same strength, and so on. And then what the sort of dotted greenish line, and what happens there is that you get a quick activation of the synapse, so a quick sort of activation of both the presynaptic neuron and the postsynaptic neuron at the same time through a burst stimulus. So it just goes, and because of this quick repeated activation, the next time you fire that synapse, the response will be higher, and it will be. Thank you very much for the mouse. That's very useful. It's a sustained, basically, elevation of the synaptic response. So a sustained strengthening of the synapse. So if we look at the next slide, we can look at the sort of molecular basis of that, and it basically has to do, at least on the postsynaptic sort of side of things, it has to do with calcium coming into the dendritic spine. If there's not a lot of calcium activates Calmodulin. We already said Calmodulin is one of those collectors. It has to collect for calcium ions. It activates a phosphatase called PP2B, among other things, and protein phosphatase to be negatively regulates AMPA receptors and negatively regulates a kinase called CaMKII. On the other hand, next slide, please. On the other hand, if we have a bigger calcium signal, the calcium still activates Calmodulin, but then Calmodulin, if there's a lot of calcium around, it preferentially activates CaMKII. CaMKII is a kinase, so it has basically the opposite effect of the phosphatase I just talked about earlier. So CaMKII increases both the number and activity of AMPA receptors. And you can imagine the more receptors and the more active receptors are in the membrane, the stronger the signal transduction can be. So this is when we say a synapse is being strengthened, it's because more and more active receptors are basically recruited. So this CaMKII is really, you know, an interesting molecule in that sense, because it really mediates this long-term potentiation of synapses. You can see the next slide, please. Actually, if we could you stop sharing? Well, I don't know. Can we try? Could you stop sharing? And I turn my video on for like the next 2 minutes, because I need my arms to explain this. And if the connection is really very bad, then we'll try to do it with the slide. So basically, CaMKII works like a Swiss Army knife. So can you see my arm? Yes. Okay, cool. So CaMKII works like a Swiss Army knife. It can open and close, and it only works when it's open. It doesn't work when it's closed. It only works when it's open because the active site, needs to be accessible, right? When it's closed, the active site is blocked. Now, calmodulin binds to basically where my sleeve would be, right? So it binds to what's called the inhibitory helix. So if nothing else binds there, then the inhibitory helix binds to the active site, and the kinase is closed. But if something else binds to the inhibitory helix, like calmodulin, then the kinase can't close again, so it remains active, basically. There's another thing that increases CaMKII activity, and that is phosphorylation at the threonine residue that is right at where the elbow would be, at the hinge, right? And again, if you get phosphorylated there, then there's a big obstacle to closing. So if the kinase is phosphorylated there, it cannot close again. And then there's a second phosphorylation site that we don't understand very well, that is basically also on the sleeve where the calmodulin binding site is, and that would prevent calmodulin from binding. Thank you. I think we can resume screen sharing. And I will stop my video again. Okay, cool. So if we can see the next slide, please. So this is what it actually looks like. So we have the active site, which is sort of the bulky thing on the left. We have the inhibitory helix on the right. We have the inhibitory helix also where the binds. We can see it's in red at the bottom of the kinase, activatory. So if the kinase is activated, is phosphorylated there, it cannot close again. And the other one shown in pink on the inhibitory helix basically prevents calmodulin from binding. So we think that it has an inhibiting effect. Okay, if we can see the next slide, please. So this is basically what I said already. So if we imagine, so an important bit of information that's missing though is that CaMKII is not just one subunit, it's 6 subunits arranged in a ring and then 2 rings on top of each other. What's important is, I think, the ring-shaped arrangement of the 6 subunits. So it's basically like a flower of Swiss Army knives. And this is important. So what would happen typically is that first, calmodulin, shown in black here, can bind to CaMKII. So here we see 2 calmodulin molecules binding to 2 subunits in one of the rings of 6. And as we said, this is incompatible with that phosphorylation site that was on the inhibitory helix. If we can see the next slide. Threonine 286 phosphorylation. This is really nice because it's a inter-subunit autophosphorylation. So that means that CaMKII, because it's a kinase, it can phosphorylate proteins, and especially it can phosphorylate the subunit that is next to itself. So a subunit cannot phosphorylate itself for sort of steric physical reasons, but if 2 subunits are next to each other, then one can phosphorylate its neighbor. But for that to happen, both of them need to be active in the first place. Both of them need to be open. One needs to be open because it needs to be active to act as the kinase, and the other one needs to be open because the phosphorylation site needs to be accessible. So what typically would happen, what we would expect to happen, is that 2 calmodulin molecules bind next to each other on neighboring subunits, thus activating 2 subunits, and then one of them can phosphorylate the other. And once the subunit is phosphorylated, it remains active and it becomes calmodulin-independent, so the calmodulin can leave again if it wants to. So can we see the next slide, please? Eventually, calmodulin will leave, and then that calmodulin binding site becomes available for the second phosphorylation. So phosphorylation at threonine residue 305, 306. That was the one that's on the inhibitory helix. And this phosphorylation then makes it impossible for another calmodulin to bind there again. So basically, the subunit is made invisible to calmodulin. And what we have thought of this as is basically an inhibition or a desensitization. So if a subunit has already been activated, it can't be activated anymore by calmodulin for a while. So you could imagine, for instance, if that autophosphorylation, the red autophosphorylation, the activatory one, even if that is reversed at some point, but the other autophosphorylation is still in place, then the subunit would be inactive for a bit longer because calmodulin couldn't bind to sort of restart the cycle. So if we can look at the next slide, please. So the problem now is that in a typical dendritic spine, there is a normal amount of calmodulin. So these are not like actual numbers, although they are not completely off. So there are kind of normal amounts of calmodulin. By normal, I mean sort of the same amounts that you would expect from any other protein. But there is a ton of CaMKII. And the reason for that is complicated. We think that CaMKII may have a structural function as well as having a signaling function. But the upshot is that you have so much more CaMKII than calmodulin that it's very unlikely that two calmodulin molecules would bind to adjacent subunits on CaMKII at the same time, which is what you would need for the autophosphorylation. So how do we get around that is basically, how does calmodulin get around that is the question. Can we see the next slide? So this is basically what, I mean, this is a very simplified drawing, right? Like we can, and I did a simulation of that in Python or something. So I just made a bunch of CaMKII hexamers, and then I released a little bit of calmodulin. Calmodulin was able to bind and unbind from subunits. And only if two adjacent subunits were bound, then you can have a phosphorylation. And in a system like that, with sort of typical ratios between calmodulin and CaMKII, you get, all your calmodulin binds CaMKII, but it's very rare that you get two adjacent calmodulins binding and this autophosphorylation happening. So if there's a large pool of CaMKII and a small number of calmodulin, you cannot really activate CaMKII. Next slide, please. So I did this in CoPaSI as well. So I had a model where I had a calmodulin, and then I showed both types of CaMKII phosphorylation, the P286, which is the activatory phosphorylation, and then P306, which is the desensitizing phosphorylation. And it kind of goes up a little bit of time, mostly because there are no phosphatases in this model, but it goes up very, very, very slowly. And most of the CaMKII in this model remains inactivated. Can you see the next slide, please? So that now, I mean, that raises the question, how can calmodulin ever activate CaMKII? Because we know it does, right? It does in the actual cell. And we can sort of think through the problem. Does anybody have an idea?
Well, it's going to be something to do with cooperativity, no?
Ah, yeah, very good. So it could be something to do with cooperativity. Any other thoughts? Because cooperativity is the coolest, we know that already. So can we? So if we had a smaller amount of CaMKII, then calmodulin could easily activate it, right? It's also to do with cooperativity, but it's also to do with relative amounts of calmodulin and CaMKII. And what we actually know is that the location of CaMKII within the dendritic spine changes as the spine gets activated. So at first we have a lot of the CaMKII sort of within the spine, but away from the active zone. And as the synapse gets activated, CaMKII gets recruited to the active zone. So if we look at calmodulin molecules, if we imagine them being concentrated in the active zone, then at first they would see sort of a smaller amount of CaMKII. And then as time goes by, more and more CaMKII would join. How does that help? Well, at the beginning, you can kind of easily see how it helps. But at the beginning, okay, let's say you only have 10 calmodulin molecules and 10 CAM molecules that can easily bind to each other and separate CaMKII. With time, as more and more
We lost you now, Melanie.
CaMKII molecules get added as you had before, right? It's likely that calmodulin. Okay, let me try and go.
Yeah, we lost you for a little bit there.
Okay.
So
okay.
Now you're back. Yes.
Okay, cool. So, so if there was like a small amount of CaMKII at first, then calmodulin could easily deal with that. And then if we increase the numbers of CaMKII coming in, I mean, again, we would have a problem as the CaMKII numbers get bigger and bigger, right?
Yeah, we lost you again.
Unless we make CaMKII molecules that are already active, basically invisible to if they become invisible to calmodulin, that's available for calmodulin binding. Is this any better?
Hey, Melanie, we are really getting very little. I mean, it's cutting a lot.
Okay, I'm trying to move around
to a better place. Yeah.
To a better place. Yeah.
Okay. We could hear you walking actually.
Okay, so maybe I'll just like stay here in the corridor and yeah.
Yeah, no, it's also cutting a little bit.
Okay, let me go to sort of the more central area.
And we're getting.
Is it better here?
Yeah, let's go on with this because we really need to get on.
Okay. Yes.
Yeah.
Sure. So basically, can you hear me now?
Yes.
Okay. So basically, what we need is a way of hiding CaMKII that's already activated from calmodulin, so that calmodulin doesn't waste time binding to CaMKII that's already being activated, and so that calmodulin basically just activates small parcels of CaMKII at a time. And the way that we can do that is actually potentially that second phosphorylation site, right, that I talked about earlier, that is incompatible with calmodulin binding. Because what it does is that it basically tags a CaMKII that has already been activated and makes calmodulin binding impossible. And we can actually look at the simulation. So if we see the next few slides. So this is just like a sort of, yeah, this is the capacity simulation. So what we did here is we added CaMKII in little intervals, and we gave CaMKII time to be phosphorylated at this other phosphorylation site before we added more CaMKII. And we can see that over time, the total number of phosphorylated CaMKII really increases. Even though at the end, right, you have the same total amount of CaMKII and the same total amount of calmodulin that you had in the previous simulation I showed you. But we get a lot more activation because we have this sort of piecewise, stepwise activation of CaMKII in little bits by calmodulin. So I have zero experimental proof that this is actually something that happens, but the model is quite, I think to me, quite convincing. And what's also quite convincing to me is that if we turn off that phosphorylation at threonine 306, so the phosphorylation on the calmodulin binding site, if you can see the next slide, if we disrupt that, then we have a similar situation to before where calmodulin cannot really effectively activate CaMKII. So that's really interesting, right? Because if we see, can we see the next slide? This brings us back to the beginning, right? So you saw a tree and you knew what a forest looked like because it was just a bunch of trees. But now, if you can see the next slide, if you look at this phosphorylation at the calmodulin binding site, if you look at only one CaMKII subunit, it looks like an inhibition, right? It looks like an inhibitory mechanism. But if you think of the CaMKII as a population, it's actually an activatory mechanism, because without this phosphorylation, you would not be able to get sort of large-scale activation of CaMKII by small numbers of calmodulin. So you knew what the tree looks like, but that did not tell you what the forest looks like. We needed a model to know what the forest looks like. Cool. Can we see the next slide, please? So this is basically the conclusions, right? More foxes doesn't mean necessarily fewer rabbits. Too much malaria can look like no malaria. A forest is not just a bunch of trees, but more importantly, right, models can show us things, can teach us things that we couldn't have maybe intuited by looking at the system just at first sight. So this is why computational modeling is really important and can really be helpful to think about a system. And of course, once we have the model, we can go back and go like, oh yeah, this is why, you know, this is why the number of rabbits increases or whatever. Then it kind of makes sense. We can argue through it, but we need the model to show us it in the first place. So I only have the acknowledgement slide left, if we can see that. So Ranjita and Kyung-Suk Pierce both worked on the proton effect, so the collector problem sort of with me. So Ranjita on the sort of theoretical thing, and Pierce made a model of malaria diagnostic testing, and Tara and Crystal Lifey both worked on the CaMKII model. So thank you very much. I'm really sorry for the connection. This is really bad. But yes, I hope you could hear some of it. Thank you.
Yes. So Les, I think we're a little bit over time, but we should allow some questions if there are now. Okay.
We can be flexible.
Yeah, I think so
Thank
too.
you.
Because, anyone has questions, please open your mic and go ahead. So I can ask a question. I'm not sure I understood completely the mechanism by which you're adding the CaMKII slowly, right?
Yeah. So I mean, I don't think the mechanism I think is unbinding from the actin cytoskeleton.
Okay.
So I didn't talk about that, but a lot of the CaMKII, when it's inactive, it's sequestered by the actin cytoskeleton basically.
Okay, well, that's very interesting because that's similar to something that happens with glycolytic enzymes as well, also in neurons.
Ah, okay.
Where lots of them have been found to be in large excess, and they're bound to actin as well.
Oh, interesting. Yeah.
And the substrates compete. They bind through some active site. I think hexokinase is the main one that does that. And then when the ligand appears, then it actually competes with the binding from the actin, and they basically get released. It's a very dynamic way of allowing more enzyme to be released when it's needed
Yeah.
without having to synthesize it because it's already
Yeah.
there.
Yeah. That's very cool. Yeah.
And this also happens in neurons, by the way.
Cool. Okay, I need to look it up.
Some very old papers, actually. Any other questions? I'm going to stop sharing here. By the way, also just a comment, the hook effect, I've also found the same thing happening actually in initiation of translation. There's a similar phenomenon that happens. And of course, there you have many different proteins that have bind together before they actually form the full complete initiation process.
Yeah.
And you get the same effect, whereas you actually, if you overexpress some protein, it actually makes the whole thing collapse. And
Nice.
you get more is not more in that case.
Yes,
More is less.
yes. Yeah, exactly. Yeah. That's a great example, actually. Yeah.
Just looking at the chat, there doesn't seem to be any more questions there. Well, I think we should thank Melanie very, very much thank you for giving this talk, even in adverse conditions. This was very interesting. And it's a very good example of why modeling is always important. I think we found out similar things already in our breakout rooms in at least some of the projects I've seen, where things don't to be obvious.
Perfect. Okay, thank you. Thank you very much.
Thank you very much.
Thank you. Bye.
Bye.
Thank you.